Great. And Chrysostome, you're welcome to start.
Okay. So hi everyone. Today's presentation I would like to give you is about demography of the frog Aubria subsigillata, biogeography, factors of rarity, and exploitation of the frog. So, amphibians are one of the most endangered species across the world, and they are being described 7,000 species of frog, of amphibian species globally. But those species are facing many threats. And these threats include habitat modification, climate change, overexploitation, and pesticides, agricultural in general. So although there is such threats, we have limited information about the biogeography of some species. Also, population trends of many another species are unknown. So this is the reason why we undertook this study in Benin, and this study focused on the species called Aubria subsigillata. His common name is West African bull frog, and it's according to the International Union for the Conservation of Nature, this species is a least concern. But although this species is least concern, we recorded in one of our studies that we undertook in 2020 that this species is exploited in southern part of Benin for food consumption. So we started by thinking about how is the demography of this species, what are information about the species in general, maybe, and to start quickly by taking some measure to regulate its exploitation in Benin. So the purpose of our study, for the first, we want to identify the occurrence area of Aubria subsigillata, then to analyze the cause behind their distribution and rarity in Benin. So this study has been carried out in two main parts in Benin. There is in the northern Benin, we sampled in the Panjari. It is a biosphere reserve of, and then in southern part of the country, we are taking some sampling in department of Weme, Plateau, Atlantic, and Zoo. In each of them, we identify many kind of area or habitat in which we could sample the frogs Aubria subsigillata. We have here a swampy area. Here we are in agricultural area. In this kind of habitats, the farmers built a kind of pond and the water inside the pond is used to water the agricultural plants. So our frogs are commonly live inside of those habitat, those ponds. So we survey inside of those ponds. Also, forest area also were considered for sampling. And sampling has been undertaken from June 2021 to May 2022. So it took 1 year. And We use visual encounter and hand capture to collect the data on our species. Visual encounter has been used because our frog do not frog in the nature, so only we can see them through our headlights. Parameters we collected smooth length. This is a measurement we are seeing here. The measure we are taking, the measure we are taking here. It is the length from the end of the muzzle to the end of the urostyle. Then also we collect that data on the body mass of the frog. And after collecting that data, also we record the information about the geographical coordinate of the frog. And after collecting all that data, we release the frog on the capture sheet. So that is this for the first. Firstly, we collect that data on environmental parameter, water pH transparency, and water transparency, ambient temperature, and ambient temperature. We also collect the data concerning the habitats. We have a fragmented habitat where there is human settlement. We have a pond, maybe fish farming, agricultural also, a different kind of activity men can practice in those areas. So we consider all those activities habitat as fragmented area. But for those habitats where we have like a forest, maybe we have limited activity of human, we consider that as non-fragmented area. So also survey were undertaken. We saw podcatcher, seller, and chief, and we submitted them to the questionnaire about for harvesting intensity and also the distribution of the place where they go to catch the frogs, the information about the place where they go to catch the frog in the nature, of course. Finally, we collected, I have always said it, we collected data on local distribution to access information, to access information on biogeography of the frog. So here is a picture of what we did, and that data measurements, environment data collection here, a frog catcher in activity where and to whom we submitted the questionnaire, of course. Here is a kind of example of fragmented area. Sorry. So for information for biogeography, we use QGIS software to map the frog distribution. For demography, program maps were used, and for threat R were used for assessing the bandwidth to analyze this data collected. And in result, we found that these frogs are only located in southern Benin, that we see it only in south of Benin. And then there is no presence, of these frog in the northern Benin. Regarding the demographic, we estimated that the population size to 12,1764.52 individuals. And then the length at first capture, which means that size at which 50% of frog we encounter in the nature caught by the frog catchers is this size is 82.88 millimeters. So more frequently, size caught is this amphibian in this annual species. Other species see individual in the size ranges from 83 to 93. And then this picture beside is showing that when the growth and the mortality decrease, the mortality per capture, which is showing by the red line, it increase. So the individual frequently capture 83 to 93 are those species who are more impacted by the rarity. And then we also that the mortality rate was higher than the growth rate, and the current exploitation level exceed the sustainable threshold, that we assess to 0.42. Also, we investigators, so we analyze the association between the variable we use for identify for assessing for the assessment of extension factor, of rarity factor, and the occurrence of the species, presence of absence of the species. And we noted that temperature, water temperature, climate type, and pH and frog harvesting was more correlative with the presence or absence of the frog Horaglanis. And I would like to remember here that when I'm talking about climate type, is that in Benin we have two kinds of climate type. We have the Guinean climate type and Sudano-Guinean climate type. Guinean climate type is a type of climate where we have two seasons, one season of long period, a long dry period, season period, and a longer raining period season. And then for the Sudano-Guinean climate, we have four types of period. We have two different short period of rainy season and two different short period of rainy season. But all of them are unequal cost. So for the variety factors, which means exception factors, of course, we identify that among the variable with data or information, the factors correlated with our occurrence, we see that ambient temperature, water temperature, and climate type was those who hold the strong scores. And then from our study, we knew that we come to some conclusion that if the exploitation level exceeding the sustainable threshold, which means that we need to quickly start by implementing a monitor harvest level. Also, to raise local awareness and integrate sustainable exploitation practice in this area, a different area we investigated. And then for the climate change, the climate type, we understand that the cause of Aubria subsigillata population decline is the climate. So we need to start by also protect this species from the impact of climate change. In some, finally, Aubria subsigillata is a species under pressure, despite the fact that despite the least concern status. And also this show data is now available to inform the conservation of this species. And also we have also to emphasize that we need to recommend inclusion in national amphibian conservation priority. This information we found from this research. Investigate also different other area in other countries in Guinean Gulf to check the information about demography, threats, and distribution of this program. Thank you for your attention. And now I'm open for your question.
Great, thank you so much, Chrysostome. That was a lovely talk. So we do need to move on to the next speaker. So if you have questions for Chrysostome, please put them into the Q&A. You can find the Q&A button a little bit to the right of the share button at the bottom of the Zoom screen. So thanks again, Chrysostome. Let's see, Lucas, do you want to share your slides?
Yeah.
Great, thank you. And we'll just wait a minute to start. Oh, there you go. And whenever you're ready.
Okay. All right. Hello, everyone. My name is Lucas Hearn. I'm currently a postdoctoral researcher at Tokyo Metropolitan University in Japan. And my current research is looking at climate adaptation in native bees and how this relates to resilience to climate change. So climate can impact ectotherms in a number of different ways. One of the biggest influences in shaping ectotherm distributions and vulnerability to climate change is the local conditions that they are adapted to. So the physiological tolerances of ectothermic species generally follow latitudinal gradients, which are also used as proxies of climate. So for instance, species in low latitudes where the climate is subtropical or tropical and generally fairly consistent tend to have narrower physiological tolerances and live closer to their upper thermal and desiccation limits. We'd therefore expect any species in these lower latitude regions to be more susceptible to any future changes in climate, such as increases in temperature. Whereas species in these higher latitude regions, these temperate subarctic regions, are expected to have broader physiological tolerances to cope with these higher fluctuations in climate that they experience, and therefore be more resilient to climate change. And one other factor that could influence climate vulnerability in ectotherms is certain life history strategies that may buffer selection on physiological tolerance traits. For instance, species that can behaviorally thermoregulate by retreating to a cool and moist nest environment, such as an underground nest or a shaded canopy, may have weaker selection on heat and desiccation tolerance compared to, say, species that can shelter from cannot shelter from climate extremes. And this could create a selection shadow that leaves these species that are able to behaviorally thermoregulate are settled to climate conditions that exceed their physiological tolerances outside of the nest or these shaded environments. And so for bees, which display really high diversity in their nesting behavior, this means that nesting strategy could strongly influence selection on physiological traits and potentially determine how vulnerable they are to climate change. So over 80% of bee species nest in underground burrows, while the remaining 20 or so percent of species create nests in plant stems or open cavities. And for these species that nest underground, these nests offer a refuge to retreat to when the foraging environment gets too hot or dry. And this may then weaken selection for higher physiological tolerances. But for species that nest in exposed plant stems and experience very little buffer between their internal and external nest conditions, this is expected to select for broader physiological tolerances to cope with climate extremes. And again, these species might therefore be more resilient to future climate shifts. And so based on this, we wanted to know really, how does nesting strategy and local climate adaptation influence vulnerability to climate change in bees? And Japan actually provides a really excellent opportunity to investigate this question. Not only does it have incredible diversity of bees with different nesting strategies, but more importantly, it offers a very large latitudinal range that covers 3 distinct climate regions. And so for this study, we collected bees across 7 locations in Japan that approximately spans a 3,000 kilometer latitudinal cline and a climate gradient that spans from the subarctic regions in the very north all the way down to the subtropical islands in the south. And at each of these locations, we collected bees from the genera Serratina and Lasioglossum. And these genera were selected because they're very cosmopolitan, they have global distributions, but most importantly, they have very distinct nesting strategies. So Serratina nest in plant stems above the ground and things like hydrangea and silvergrass, whereas Lasioglossum bees construct nest below the ground. Additionally, the size range of species within these groups are also comparable. But most crucially, representatives from each genus can be found at all of our sampling sites throughout Japan, and that made them ideal candidates for this kind of study. So to determine the vulnerability of these bees to climate, we used a combination of thermal tolerance assays and desiccation stress assays. So we performed critical thermal maximum and critical thermal minimum assays using a water bath that was heated or cooled by a thermoelectric Peltier module connected to a temperature controller. And so the bees are essentially placed in this water bath and then ramped at a rate of 0.2 degrees per minute. And this gave us measures of their upper and lower thermal limits at each location. And we also performed desiccation experiments by placing an individual bee in a desiccation chamber with one half filled with silica gel beads, which created a desiccating environment for the bee, which was placed in the second half of the chamber. And we also weighed this bee, each specimen before being subjected to this environment and then after to get the water loss and also to be able to calculate water loss rates. And so at each site, we also placed data loggers at nesting sites for the ground nesting bees, so in the ground, and also stem nesting bees. And this allowed us to detect nest microclimate differences at each location. And to get a more broader view of the annual climate conditions, we used the climate data from the National Centres of Environmental Protection. Prediction, sorry. And from this, we got temperature and relative humidity data. And we used both of these to then calculate a third metric called vapor pressure deficit. And this is essentially the drying power of the air. So it provides a much more biologically meaningful measure of evaporative stress faced by ectotherms. And this climate data was then validated against weather station data at each location in Japan. And so we used temperature data from this to create a mechanistic niche model using the R-package niche mapper. And this allowed us to generate hourly operative foraging temperatures and also temperatures for both stem and ground nests at our collection locations. And we also used this operative foraging temperature that we calculated to calculate thermal safety margins based on the difference between species CT max, so critical thermal maximum, and the operative foraging temperature. And also used this projected temperature data for the years between 2081 and 2100 to calculate a future thermal safety margin. And so as we can see from this climate data, basically it shows this nice trend of climate variation across latitude and across our sites. So in the very north, you're getting very variable climates throughout the year, and especially in this spring to summer period, so April to October, whereas more in these lower latitude subtropical regions, you're getting very stable climates. And we might therefore expect bees in these lower latitude subtropical regions to have narrower physiological tolerances, given this low climate variability. Another way we wanted to look at climate variability is within these nesting environments. So our modeled nest microclimates show that stem nesters experience a lot more variability in climate compared to ground nesters, as you'd kind of expect. And this was also reflected in our actual data loggers that we placed in the nesting sites. And so these examples shown are just for our Tokyo site to give you just a feel for that. And so for our results, what we found was basically that stem nesters did have much broader thermal tolerance in general, but actually we didn't find any difference in CT max across latitude. However, we did find that CT min, so lower thermal limits, were decreased with increasing latitude. So the higher latitudes had much lower CT min, And yeah, especially in subarctic regions, they had much lower CT min. And our thermal safety margins also showed a trend across latitude that you would kind of expect. So much lower in these subtropical regions. And generally ground nesters had a lower thermal safety margin compared to stem nesters. And that trend was very similar for our future projected data, except kind of flattened out across latitude a bit. And so these findings are supported by previous studies that also show that CT max in ectotherms doesn't actually vary across latitude, but the thermal tolerance breadth or the patterns you get across latitude are actually due to these differences, this variation in CT min. So you're getting much lower CT min in these colder regions, and that's the trend you get across latitude. And for our thermal safety margins, this is also in line with the climate variability hypothesis. So basically that there's much lower in our in lower latitude, our subtropical regions compared to our subarctic regions, much higher, suggesting that bees in these regions live closer to their upper thermal limits, but also that bees in these regions may be more vulnerable to climate warming in the future. And so we also found strong patterns in desiccation tolerance across latitude, but also a high differential response between stem nesting bees and ground nesting bees in our study. So stem nesting bees had far higher desiccation tolerances in general. We found that latitude significantly increased the time to desiccation in our desiccation chambers across both bee groups. But the rate of water loss across latitude was only had an influence on ground nesting bees, so significantly higher in our subtropical low latitude regions. And when we actually relate this back to patterns, our vapor pressure deficit metric, we found that water loss, the rates of water loss was highest in locations with the highest average annual vapor pressure deficit. However, this is a pattern that actually contradicts previous hypotheses that high desiccation resistance should evolve in high vapor pressure deficit environments. But actually, when we look at the annual variation, which we calculated as the coefficient of variation for vapor pressure deficit rather than annual mean vapor pressure deficit, we found that it more strongly correlated with the expected patterns in water loss. And this kind of gives you, looking back at this environmental data, you can kind of see that in these higher latitudes, you're getting a much higher max VPD, much higher variation in VPD compared to these low latitude regions. So bees in these higher latitude areas are having to deal with higher maximum or like higher extremes of VPD compared to the more stable subtropical regions. And so essentially the two biggest takeaways from this study so far have been that firstly, that desiccation stress is a critical component of climate vulnerability that often goes understudied, but especially in ground nesting bees, it seems to affect them a lot more than thermal stress, and can even be high in regions of high humidity, such as subtropical regions. And secondly, that's the species best equipped to buffer themselves from environmental extremes, such as ground nesting bees that can behaviorally thermoregulate, may actually be the ones that face the greatest risk of extinction under future climate change scenarios. So if bees have the ability to behaviorally thermoregulate, they may have evolved weaker physiological tolerances that make them susceptible to changes in their foraging climates. And thank you for listening.
Great. Thank you so much, Lucas. Appreciate a great talk. In the interest of time, we'll move on now to our next speaker, Nikos. If you do have questions for Lucas, please feel free to pop them into the Q&A at the bottom of the screen. Thanks again, Lucas.
It's fine. You can see my screen and hear me and everything fine.
Absolutely. So we'll just wait a
Yep.
few seconds for 11:45. Great. Whenever you're ready.
Sure, I'm ready. Okay, so hello, I'm Nikos. I am a postdoc at the University of Turku, but I will talk to you today about the last little project I did during my previous postdoc in the Max Planck for evolutionary anthropology. And it's a little descriptive project, so my presentation is mostly a discussion. It has less results than usual, so please bear with me. And it's of course about the post-reproductive lifespan in gorillas, in mountain gorillas. So post-reproductive lifespan represents a paradox because it's not clear why individuals live past their age of last reproduction. What do they, what is the benefit for it? And in the recent years, there are several studies on that. Of course, I guess you know that post-reproductive lifespan is it's a human trait, but it's not so common among mammals. Was a recent comparative analysis in toothed whales showing that there are some species which exhibit menopause or post-reproductive lifespan among these species, but this is the family with the most, the taxa with the most common, where this trait is most common. And 2 years ago, there was also one study showing menopause and post-reproductive lifespan in wild chimpanzees, specifically in Congo chimpanzees, in specific populations of chimpanzees. In this plot, you can see the oldest females of the population, and with the yellow dots represent the beginning of the study period, let's say of a female. Blue is the end. Red dots are reproductions, like births essentially. And you can see that like for several years, more than a decade, in many cases, females do not reproduce. So they are characterized post-reproductive. I will define below what means post-reproductive. In this same paper, people also use the hormonal data, analyze the specific hormone, which is reflects the drives ovarian function and cycling, essentially. And they found that the levels of this hormone for post-reproductive females are similar to females who are experiencing lactational amenorrhea, and much lower than females who are cycling, which are like hormonal evidence that indeed there's a physiological change in these old females. In gorillas now, so on the right side, you see a gorilla study, which on the top you see like the cycling of this same, like the levels of this same hormone in nulliparous females. You can see that the levels are very low, like here. I'm not sure if you see my cursor. On the bottom, you see, in the middle, you see mature adult, let's say prime age females. So there's a periodic increase decrease of this hormonal data of this hormone, sorry, not data, of these hormones, suggesting the cycling, so ovarian activity, ovarian function. And at the end of the bottom, you see data from only one female, unfortunately, which is around 38 years old, and you see again that the hormonal levels are more similar to the nulliparous females, suggesting that this female does not really cycle. So now we use data from. I would like to first thank Martha, who was my advisor, and she's the director of the gorilla projects in the Max Planck, and she's maintaining two field sites, one in western Gabon of western gorillas. I'm not talking about those today, but it's like my default slide talking about the project. I'm talking today about the second field site, which is in the southwest Uganda, studying mountain gorillas, specifically Bwindi mountain gorillas. The name is from the National Park Bwindi. There's only one more population of mountain gorillas in the wild, which you might.
Nikos, we've lost your sound. Or at least I have. Is that the case for others?
Yeah, he's not audible.
Now you can hear me again?
Yes.
Yeah, now you're audible.
Okay. Sorry. Where? Okay, I will try to.
You had just finished talking about the last population of Western mountain gorillas. Yeah.
Okay, so briefly, I'm not talking about Western gorillas today. I'm talking only for the mountain gorillas. This is just the default slide about the project. So on the right, on the east side, mountain gorillas, Bwindi mountain gorillas, and there's only one other population of mountain gorillas, from which the previous study comes from, the study that showed the hormonal levels. Okay, so these are just pictures to show you like that how females and males look. Males are not relevant, just to show like how very old males look. So on the left side, in both cases, we have like a prime-aged individual, a female and a male. On the right side, we have like very old individuals. And I have to note that this population of gorillas is the ones with the largest, the highest lifespan, with a very small difference, but still. So it was an opportunity for us to study this question here. Okay, so we have data more or less of 30 years. I'm saying more or less because we have like detailed data since behavioral fine-grained data since 1998, but we have knowledge of the population from the previous years that was like during the habituation. Now, as I said, I will define here what's post-production. So post-productive females are those who live past the age of their last reproduction for longer than the mean plus 2 standard deviations for successful interbirth interval. In our case, this was 7.7 years because we have a 5.1 average interbirth interval plus 2 times its standard deviation, which is 1.3. Okay, so females who have lived more than 7.7 years after their last birth, we can call them post-reproductive. Here you can see all the females in population. On the left is their ID. On the right, you can see the number of years since their last reproduction. Red is the beginning of the study. I mean, their entrance to the study, if I can call it like this. Black is the end. So basically, it's either when they have disappeared or died, or when the study ended a few months ago. And the little gray gorillas are births. Okay, so you can see that we have 7 females who have lived more than 7.7 years after their last reproduction. 6 out of them, they're older than 35 years old, which is the maximum age we have observed a birth of the oldest female. And all the 7 post-reproductive females have lived more than 10 years. So they have and given that females very rarely live more than 50 in the wild, or even in captivity, and they are adult more or less at 10. From this maximum 40 years, if let's say they have a maximum adult lifespan of 40 years, if we have 10 years of post-reproductive lifespan, means that more than 25%, or at least 25%, they can spend it as post-reproductive. Okay, so now we'll go by one in the main hypothesis that have been posed about the evolution of post-reproduction. So the first we have the reproductive conflict hypothesis, according to which all females cease reproduction to avoid competition with younger females, young related females. And there are evidence for that in killer whales. You can see here that when a younger and an older generation females give birth at the same time, the calf of the older generation mother has a lower survival probability. So it's the blue line in comparison to the green. But in gorillas, females exhibit secondary dispersal, meaning that they are living, they can go in a group, they can leave an adult group, go in another group, reproduce, and after live again. So the local relatedness is likely small, smaller at least than the average local relatedness is smaller than in killer whales or in other species which are strictly female philopatric, meaning that they are unlikely to compete with their daughters for reproduction, because the daughters may have left the group, or themselves may have left the group. A second hypothesis you might have heard of is the grandmother hypothesis, according to which all females gain greater fitness by helping their offspring to reproduce, their daughters to reproduce, or their sons to reproduce, than reproducing themselves. So it's better to divide their help in all their different offspring for them to reproduce than concentrate all their effort to their own one new offspring, which is of course more demanding. And we have evidence for that in humans. But again, in gorillas, both sexes disperse, meaning that females are unlikely to live with their grand offspring, because their offspring have probably left the group and have reproduced elsewhere. Okay, another related hypothesis that might make more sense in gorillas is called mother hypothesis. So here, females cease reproduction to maximize investment to existing offspring. This makes more sense in gorillas. Why? Because not only gorillas, in all the great apes like hominids, maternal presence is important. Care and support is important even for juveniles and even for adults, if ever you live with your adult offspring. There are studies on that. And just an anecdote that my advisor Martha has pointed out, that when females cease reproduction, then they're old, they look, whatever this means, better to us, at least to observers, than females who have given birth. I guess we have like mothers in the audience, or even maybe fathers, but mothers, I think, more. They are not surprised by that they're, they're really, yeah, that it's like a struggle, essentially. Okay, so a last hypothesis, named antagonistic pleiotropy hypothesis, suggests that reproductive cessation is not adaptive, but occurs because due to like selection shadow that I think the previous speaker also mentioned, the phenomenon. So here the idea is that when we have genes that have like double effects, and the positive effects are expressed in early life, and the negative effects in later life, but individuals rarely live at these late ages for these negative effects to be expressed. There is a selection shadow over these negative effects, and therefore these genes are not purged from population. So when, for some reason, we have individuals reaching these old ages, we observe the negative effects, which here would be the reproductive cessation. Okay, so in the study of chimpanzees that I mentioned in the introduction, the menopause is documented in the population of wild chimpanzees with the highest lifespan, the highest survivorship. Anyway, the females in this population live longer than females in other populations. The red line is the Ngongo, is the population that the menopause has been documented, and blue is the average of all the other several other wild chimp populations. In gorillas now, in Burundi, as I said earlier, these gorillas are the ones who live a bit longer, and they live in an environment where they don't have natural predators anymore, because leopards, which is the only non-human predator, is not there, and there are also extreme, let's say, conservation efforts. So indeed, there might be that these animals live in more favorable conditions, at least from the conditions we have been observing the previous years. We don't know in the historical, of course, past, in the long past. Okay, so menopause. Can we use the word menopause? I would say not yet, because I mean, respecting, of course, the people of, there is a lot of debate when is the menopause, if like we need really hormonal data. So it's not clear that's menopause, but I think we have three good reasons to believe that it might be indeed. So the first is our results, showing that females can live up to a decade with no reproduction, and they're old. They're like more than 30 at least, and 35 in most cases. Second and third, sorry, I put them together. So first on the bottom left is this. Oh, that's the wrong plot. Okay, so the second is like the one female I showed that it's like 38 years old and she was not really cycling like a newly parsed female. And this, what I'm showing here by accident, is the actual female who is the actual cycle. How would it look? And the final thing is that from studies in captivity, we know that 1 out of 4 females who are more than 30 years old, that's from Western gorillas, but whatever, these females do not cycle. So we think that it might indeed be menopause. I would like to thank everybody, like all the people in the field who collected the data. Martha, again, you for your attention, and I'm done. And thank you.
Great, thank you so much, Nikos. We have time for one short question if anybody has one. So yeah, I mean, I think you got to it at the end, but what would be the sort of next key piece of data that you'd want to gather here?
Yeah, it would be great to have hormonal data, of course. But we don't for the moment. Martha is, I mean, trying and would like to have this data one day. Yeah, so we are limited. That's why don't have so many results to show. We have like the main observation is that we have females who live longer than in other populations, but it's not only. I'm not saying that it's because it's a by no. How can I say? It might be because observing great apes in the wild is hard. It's like you need to observe, especially post-reproductive lifespan. You need to have at least a few decades of data. So you need patience and ideally hormonal data. And I think these two things.
Thanks so much, Nikos, for a great talk.
Sure.
Okay, so we will now move on to our next speaker. Akhil, if you'd like to share your slides.
Yeah, okay.
For Akhil and Akanksha, I don't think you were here at the beginning, so when we're at 12 minutes, I will put up a green check mark. And when we're at 14 minutes, I'll put up a red X just so you know. Okay, so Akhil, you can get going whenever you're ready.
It's my slides.
Yes, you're good.
So hello, I'm Akhil Sadiq. I'm a PhD student working with Ulla Ramaya at ISA Thiruvananthapuram. And today I'll be talking to you about a part of my PhD work that I'm doing. So the title of my talk is Decoupling Detection and Recognition in Animal Camouflage. So animals have these fascinating variety of strategies that they use to hide in plain sight. But some animals have colorations that match the background, and we call this as background matching, where others have these high contrast markings that break up their outline and shape. We call this as disruptive coloration, while other animals just straight up mimic objects that is of no interest to their potential predators. So all these strategies and many more come under what we call as camouflage. Here, I'll be focusing on disruptive coloration, where animals have these high contrast markings along their edges, which kind of breaks up their shape, and also on the surface, which breaks up the surface of the animal, making it harder to be detected. And where other animals can have markings which are not at the edge, and it's rather limited to the surface of the body. The first one, I'll call it as outline disruption. And the second one, I'll call it surface disruption. So one of the most common experimental paradigm to test for camouflage is using human beings, where we'll have human beings that are tasked to find targets in a computer game, sort of like Where's Waldo kind of scenario. And what studies have found is that when disruption is limited to just the surface of the animal, they're easier to detect, to be detected, or people take lesser time to detect those targets compared to when the disruptive coloration is also found in those outlines. But when you think about it, for a predator to successfully capture a prey, first have to detect its location of the target and then recognize it as an edible object. But almost all of the human experiments have this assumption that if you know where something is, you know what it is. And such an assumption could be, for lack of better words, dangerous. So I asked this question, are detection and recognition coupled? So basically, if we have two targets, and if a pattern can make one target harder to be detected, can it also make the target harder to be recognized? So to test this, I used human volunteers to play a computer game where I had such grass backgrounds and I extracted two most common colors. And using these colors, I created three prey types. One was outline disruptive, where the outline and surface is disrupted, and two, surface disruptive treatment, where the outline is left intact. And each prey type had three different shapes: a lion, tiger, and a wolf. So I went around my campus recruiting volunteers and I asked them to play a game where they first have to detect the target. And once they detected, I record the time as detection time. And once detected, they were asked to figure out the shape of the target. And the time they would take to recognize the shape would be considered recognition time. And we did the analysis and found that people took longer to detect a target whose surface was disrupted compared to when the outline and surface was disrupted. But this tendency was reversed when it comes to recognition time, where they actually took longer to recognize targets where the outline was disrupted. So this kind of made me conclude that detection and recognition are not coupled and they might at least be partially decoupled. So I proceeded and I asked the question of where this outline should be disrupted. And if you look at these animals, these are all putatively disruptively colored and the markings are not limited to, not found randomly across the body, but are rather limited to different regions. So if you consider such an animal with an outline, it could either have disruptive markings running randomly across the entire body, or the disruptive markings could be on these diagnostic regions of an animal, which we call as features. So I asked, should disruptive coloration, disruptive features. So to answer this, I came up with these three shapes. And what's peculiar about these shapes is that they share a common body part, and the only difference is in their head. So the head here is the feature. Using this paradigm, I created four treatments. One where the entire body was disrupted, one where the common part was disrupted, and one where the feature was disrupted, and one where none of the outline was disrupted. And similar to the previous experiment, there were three shapes, and we ran a similar set of experiments, and we found that detection time was higher when the common part was disrupted compared to when feature was disrupted. However, when it comes to recognition time, it was higher when the feature region was disrupted. So this led me to conclude that disrupting features is what impedes recognition. So yeah, with this I end my presentation, and I would like to thank all these wonderful people and Evolution 2025 for giving me this opportunity. Thank you.
Great. Thanks so much, Akhil. So we have lots of time for questions. Does anybody in the audience have a question? So the last result, right, where you're showing that disrupting what you called features maybe has an outsized effect. How would that translate to some of these real world examples that you gave, right? You gave these three examples, the panda and the bird and something else, where you noted that the disruptions aren't uniform. Like what would you consider a feature? How would you distinguish a feature from a non-feature in a real
Yeah, that's a really good question and a really difficult question to answer. So that's the next part of my work, which I'm trying to do is figuring out animals right now focusing on butterflies, where we look at different parts of the wings and seeing if a certain region of the wing has more disruptive bands compared to another part. So right now, besides very few experiments with frogs where they have disruptive bands running across their legs, which could be a feature. We don't have much idea about what actually is a feature.
Okay, yeah, that makes sense. Thank you. Because the other thing I was wondering about, so you've got these two things now, detection and recognition. I mean, it's hard to tell from sort of a handful of computer game experiments, but they're not coupled. So, do you have a sense of what the better investment is? Like, should prey items or whatever be focused more on detection or recognition, or either is fine? Do you have a sense?
I think the priority should be to evade detection rather than recognition. But then again, it depends on, let's say we have this animal which has two different backgrounds in its habitat. Let's say a grasshopper can match which has can also be in grass and in, let's say in sand.
Yeah.
And it matches grass but still in sand it might be misrecognized as part of the grass. That's what I think.
Okay.
Yeah, and if you look at certain predators, they do rely on being detected but not being recognized, like aggressive mimics. So in those scenarios, it would be better if you could get yourself detected but still not be recognized.
Okay, great. Thank you. So I got a question for you. I got it in the chat, so I'll read it out for you. So this is from Arjun CK. So I'm curious about the hypothesis, as in a real world scenario of invertebrates, there will be multiple sensory organs coupled and working for the same detection and recognition, right? So
Yes.
Yeah.
So here my focus was on the vision aspect. Focus on vision, but yeah, definitely there are auditory aspect to it where one good example would be with moths and bats, but I'm not really in the position to discuss that.
Okay, thank you. Other questions either in the Q&A or chat or out loud.
Yeah. Do you consider shadow? Okay. So in these games, we have not tested the shadows because these were computer games. But in my another project, we did check the effect of shadows using the chickens, trained chickens, and we figured that shadows do play a part, and there it was not as much as matching a certain background, but rather hiding the shadows. Like being in a darker background is always beneficial. Shadows.
Other questions for Akhil? Okay, well, thank you so much again, Akhil. So we've got a few minutes left, but we will wait until 12:15 to start the next talk. Good, we can see.
Are my slides visible?
Your slides are visible and we can hear you. We'll just wait a few seconds until 12:15 on the dot.
Sure.
All right, whenever you're ready.
Hello, a very good afternoon to everyone present here. Thank you for the organizers of Evolution for giving me this opportunity to present part of my work. So today I'll be sharing a slight insights from a project I'm working on. I'm a PhD scholar from Shiv Nadar Institute of Eminence, India. The broader area of my research objectives in understanding the genomic adaptation in subterranean fish, which are found in the lateritic aquifers of Kerala, India. So, starting with a brief introduction towards no, it's not working. Starting with a brief introduction about the model organisms around which my entire research revolves is this a beautiful subterranean fish known as Horaglanis krishnai, and this fish is the first blind catfish discovered from India. So it is a subterranean fish which has lost its eyes, and it lives in its unique habitat of lateritic aquifers of Kerala, India. It has reduced its pectoral fins. It has lost its eyes. It's completely blind, and it's so transparent that you can see that this is a blood red color appearance on this fish. So, and it's very small in size to pass through these pores of the aquifers. So these are the unique morphological adaptations which are found in this blind catfish. So when we started this project, so we thought that we should understand adaptation in terms of its whole genome, not in terms of some specific particular objectives we should consider in our mind, and we should do things. So what we started is to study the genomic adaptations. The first thing which we started is with the mitochondria, because this particular fish, as I told, that it lives in the aquifers of Kerala, and these aquifers, the habitat is so unique and fascinating that this is not accessible by humans. So they live deep down inside the caves, which are oligotrophic in the environment. So they do not have plants, so they are totally dependent on the food which the water which the water brings in. And also because, as I mentioned, that the aquifers are lateritic in nature, so they have a lot of iron in them, and the pH is also low if you compare it to the surface ones. So this unique environmental conditions in which this organism lives, we wanted to understand that how, under such circumstances, how the entire this genome is helping these organisms, these subterranean fishes, to evolve in these environmental conditions. So first, what we thought is to see about the mitochondria. The reason to think for the mitochondria first is because mitochondria is the powerhouse of the cell. So if there is a lot of iron in it, and there's a pH also low, so that is imposing a lot of some stresses on the mitochondria. So mitochondria each second has to combat those stress. So to live a normal life in those natural habitat, they must have evolved certain beautiful adaptations. So we wanted to see first the mitochondria, how it is the mitochondria has evolved in response to it, in response towards its adaptation to the environment. So starting with my hypothesis, we thought that if the environment is not the similar like the surface normal catfishes have, then there must be a relaxation in their selection. We know that there is always a natural selection on the genes which are there. So first of all, that selection needs to be settled down. It needs to be relaxed, so that the genome can accumulate more mutations in it, which are adaptive in nature, so that it can help to diversify. And that's what we started when we started this particular project. For the talk which I'm starting today is I'm only presenting a part of my work, which is towards the mitogenome annotation, assembly, and the molecular selection analysis of this particular beautiful blind catfish. The methodology which I followed is, first of all, I assembled the mitogenome of Horaglanis krishnai using two sequencing platforms, MG and Illumina. Short reads were taken. They were assembled using a software known as MitoZ, and it was annotated using another software, Mito Annotator. The structural annotation was done using another software, MitoS, which annotates the tRNA and the rRNA. And the circularization of the mitogenome was done using the software Chloroplot. Now, for studying the molecular selection pressure analysis, I took 112 siluriformis fishes, and the data was taken from the NCBI. All the protein coding genes, they were splitted using a software called as eMito or EZ-split. There were two things which I did when I took these 112 siluriformis protein coding genes. First of all, I took a subset of 75 species, because I actually used DataMonkey server. So the DataMonkey server can take up only 75 species. So I took from 112 listed species, I took 75 of them, and using 3 different tools, relaxed, fell, and busted, I checked the molecular selection pressure on these 13 protein coding genes of these 75 species. Another thing which I did is, for all the 112 celluliformous fishes, I tested the histidine content for those 13 protein genes of mitochondria. Why I check for this histidine content, I'll be talking a little later in my slides when I'll go through. So there were 3 tools which I used for the molecular selection pressure analysis. It was first the relax, which will tell us that how much there is a selection on these particular protein coding genes. Is it being relaxed, or is it still being there? Or then I checked for the FEL, which test for the which will tell that how much there is a positive diversifying sites in the codons of the gene. I also tested for the busted tool. Use the busted tool to see whether there is an event for positive events of positive episodic diversifying selections in these mitochondrial protein coding genes of Horaglanis krishnai. The results were amazing, as expected. We were thinking of, because it belongs to its environment is way different, so we were expecting beautiful results out of this particular blind catfish. Apart from the mitochondrial statistical analysis, which is like, it has 16,526 base pair, its GC content is 42%, and the rRNAs are 2, the 12S and 16S. The 2 important findings which we got from this mitogenome assemble annotation was that you can see there is a duplication of histidine tRNA. So there are not 22, there are actually 23 functional tRNA histidines which are present in the mitochondria of this Horaglanis krishnai. And another finding which came out from this mitogenome work was that there is a loss of this ND2 gene. So there are not 13 which we think for a mitochondria, but there are rather 12 functional protein coding genes of Horaglanis krishnai. When I got this result, I was amazed. So first of all, I thought to check that why there is a duplication of histidine tRNAs in this mitogenome of Horaglanis krishnai. So for this, first of all, what I checked that is this duplication is because of the gene rearrangement which we see in other organisms also. There are various papers which are published in other organisms which show that this duplication events occur because of these gene rearrangement. But when I saw the gene arrangement of Horaglanis krishnai, it was exactly the same as for the all other 112 all other celluliformous fishes, telostats fishes rather. Then I thought that is this duplication event is in response to the histidine content, and that's why I checked for the histidine content of all 112 species taken of celluliformis, that is this there is an increased demand of histidine in the mitochondria in its protein coding gene with respect to its environment. So but the results were not that way. The Horaglanis krishnai, we can see that this violin plot shows that it has in its every protein coding gene, it has its all histidine requirement as normally it's for the other celluliformis fishes. Then I saw the structure of both the duplicated tRNAs present in the mitogenome of Horaglanis krishnai, and we can see that the structure of both the tRNAs is identical. So there is only one change at the penultimate 3rd prime position of this duplicated tRNA, where this A has been replaced by G. Other than this single mutation, there is no change in the structure of both the tRNAs which are there in the mitogenome of Horaglanis. So we hypothesize that this duplication is it's just not a mere event. It's being actually selected and preserved by the nature in the mitogenome of Horaglanis krishnai, which is in response towards its environment. As I mentioned that this fish lives in lateritic aquifers of Kerala, and these aquifers are rich in iron content because of the lateritic rocks. So obviously, this is my hypothesis right now. I don't have a particular answer for this question that why there is histidine duplication in the mitogenome, but we are assuming that This is not a redundancy in this mitogenome. Because of the environment that it has more of iron and low of pH, histidine, the only amino acid known to have a buffering capacity, might be this is in response towards that particular environment. So I'm still figuring out that why there is this duplication event in this mitogenome. But this is a very beautiful result which we are showing for the first time in a fish, that there is a duplication of histidine tRNA. Then there was another very important finding from this particular work, that you can see that there are so many stop codons at different points in ND2 gene, and so many stop codons in the entire gene sequence of Horaglanis krishnai, making this ND2 gene lost its function. So it's present as a sequence in the mitogenome, but it's not functional. So we can imagine that mitochondria, who is an organelle of the known as a powerhouse of the cell, and ND2 gene is playing an essential role in that complex one. So then how this particular loss is this Horaglanis fish is combating with this loss. And also to be mentioned here that this functional loss can only accumulate when there is a relaxation in selection. When the selection is relaxed, only then so many stop codons can be inserted into a gene sequence. And so third step which I moved towards is for the molecular selection analysis for all the genes of the mitochondria of Horaglanis krishnai from ATP6 to ND6. For all the 12 functional protein coding genes, as I mentioned, I took 75 celluliformis species, and I checked using 3 different tools that is there any relaxation. So for Horaglanis krishnai, we can see that out of 12 functional protein coding genes, 10 are under significant relaxation, which it's way amazing to understand in terms of in terms for organelle like mitochondria, which is known to have a strict kind of a negative selection, that its genes are so much under selection that they're not allowed to relax so much. But this is the first time we'll be reporting that in a blind catfish, there out of 12 functional protein coding genes, there will be 10 which are showing significant relaxation. And our result also is supported by 2 different other tools also, which is FEL and busted. Busted tells that is there an event of episodic diversifying selection. So Horaglanis krishnai is showing in 3 genes out of those 12 that there are events of episodic diversifying selection. And also the result is supported by another tool, FEL, and we can see that in all those 12 functional protein coding genes, in 10 of them there are so many sites which are showing that there are diversifying positive selection. That means they're accumulating more of mutation, which is helping this fish to combat and adapt towards its natural latratic aquifer conditions. And only these mutations can help this fish to evolve and adapt it better to its environment. I didn't stop over here. So what I did is I also checked and compared among different other catfishes also. So what I did is I selected 3 other catfishes which are not from India. So it's Pretilia, Ferretophila, Trogloglanis petersoni, and Zanbergus dorsalis. So these are also subterranean fishes. And Horaglanis krishnai is my one. For comparison, I took 3 other surface fishes as well, and we can see that it's only the Horaglanis krishnai which is showing 10 protein coding genes which are relaxed in the selection, and none of the surface fishes are showing any kind of relaxation. And that's what our hypothesis was, even that if you go in a different environmental condition, then there should be a relaxation in the genes.
Sorry,
And
Akanksha, you should be finishing up now. We're getting very close to the end.
yeah,
You're the
sorry.
last speaker, but we
Yeah,
should.
I'm just winding up. It's my last slides next. So then it's the Horaglanis krishnai only, which is showing so many genes which are relaxed, and even it is only Horaglanis krishnai which has 3 genes which are showing busted results, and even the maximum sites which are under diversifying positive selection in case of Horaglanis krishnai. And the comparison between other subterranean fishes and the surface fishes are also towards our hypothesis only. moving towards my conclusion only, that it's the basically the mitogenome of Horaglanis krishnai is unique among the other catfishes taken. It has duplicated its tRNA histidine and there's a loss of ND2 genes. And this all event is basically, in a one line, if I should say, that there is a gene duplication, gene loss, and importantly, the relaxation and selection, which is leading for this diversification of several codon position, making the mitogenome of Horaglanis krishnai unique among the other catfishes, and thus making it an excellent model for understanding the effect of subterranean niche on mitochondrial evolution. Lastly, I just wanted to thank, first of all, the organizers of this conference for giving me this platform to present just a part of my project and give some beautiful insights for this beautiful blind catfish from India. And also my supervisor, along which this project is running, and my lab mates and the Shiv Nadar Institute for giving me funds for this project.
Great. Thank you so much. That was a great talk. And thanks for everyone in the session. I really enjoyed it. I can leave this session open in case folks have questions, either aloud or through the Q&A. Otherwise, we'll see you this afternoon. Thanks, everyone.
Thank you.